Okay, hello, SCIE193. So Prof. Braden Evans checking in with you guys for week 13. We had some disruptions this week, and so I'm providing a very abbreviated and short version of our lecture through this recording rather than our standard live seminar. Of course, we'll be back to our regularly scheduled programming next week. All right. So this week we're focusing on developmental biology, and while I understand that the recorded kind of screen interface is nothing like in person in the classroom, I'll try to compensate for that by keeping this as short and sweet as I possibly can. Okay. Now I've got a good list of terms to know this week. Not so much like maybe last week and next week where that wasn't as much of a priority. In this case, there's probably like a good number of new terms, so it's worth going through some of our terms to know real quick. We'll make reference to haploid cells, which are cells that have one single set of chromosomes rather than the two sets that we have in all of our body cells. Here we're talking about egg and sperm cells. Okay, so we're talking about developmental biology today. We'll talk about fertilization and that sort of thing. And so sometimes we're referring to the sperm or the egg. Well, those are actually entire cells. The egg in particular is a huge cell. It's a single cell, but they only contain half of the genetic material for an organism. Somatic cells, those are the regular cells, okay? So those are the cells that compose our body, okay, the ones that make up our body. And in this case, we're talking about cells that would have, two copies of the genetic material, right? The diploid zygote, that's when our sperm and egg cells fuse together during fertilization, okay? And that's the result is a zygote, the formation of a zygote. And just another term for our sperm and egg cells that you might see is the term gametes, okay? So gametes are sperm and eggs. These are cells, again, that are haploid, right, with half the genetic material. The zona pellucida, this is like a protection layer around the plasma membrane of the egg cell. All right, it helps to protect the egg, but it's also where some of the fertilization action takes place. An embryo is the very beginning, early stage of development. And so after we have this fusion, right, this fusion of the egg and the sperm, we end up with a zygote, and shortly after this develops into an embryo, right? The acrosome reaction, that's also part of the fertilization process, okay? So the acrosome is actually this organ that covers the head of the sperm cell, and it contains the enzymes that help break down some of the material that helps it sort of penetrate the egg cell. So it helps. This is an important process to help facilitate fertilization, the fusion of the gametes into a zygote, right? Um, Telomere shortening. So then we'll move on. We'll talk a little bit about cell death and things that happen later on in the developmental process. So when our cells are replicating, they replicate a lot over time. You get this duplication of DNA, right? DNA starts to make copies of itself so that cells can reproduce. Well, over time, this process sort of decays a little bit. So you end up with shortened DNA molecules. These are the telomeres, are these strands kind of on the ends of DNA molecules. And over time, as cells begin to age, those strands begin to shorten. We also have something called oxidative stress. These are these O2 molecules that we have in our body, oxygen, but these ones are a little like hyperreactive, I guess you could say. And if you were to draw this out, it would be O2 with an asterisk up to the top right, okay? And that is what we call a free radical, okay? And so free radicals are this hyperactive oxygen molecules that sort of break down different elements in our body. And so that's sort of one of the things that contributes to cancer. So oxidative stress is something our body combats, okay? And one of the ways we combat that is through apoptosis. And so this is like pre-programmed cell death. It's genetically activated, and it sort of helps to fight against cancer and the proliferation of like mutated material, the proliferation of cells that we don't want, right? Cells that are defective. Apoptosis helps to kind of check that by giving cell lines like a certain shelf life before they start to die off, and we kind of focus on the reproduction of newer cell lines. Okay, so we took a few minutes to go through those key terms because a lot of them are probably new to you or newish, or you haven't heard them in a while. So now let's get into our unit, okay? So we're going to talk about, a lot of the sort of beginnings of life, the beginning of organismal development, particularly in organisms like vertebrates. And then we're going to close off with a case study where we just kind of talk a little bit about insect development as well. So the invertebrates or vertebrates, obviously, an important consideration, right? Excuse me. So let's get through it. So gametogenesis. If you recall, I said gametes. That's another word for haploid cells. It's another word for sperm and eggs, right? So these are those cells with the half the genetic material that fuse during reproduction, okay? So gametogenesis, that is like the development, right? The formation of the different gametes. So in males, it's spermatogenesis, okay? So in the testes, you get the production of sperm, and sperm are individual cells that are mobile, right? They move and they swim towards an egg with the goal of fertilizing it. And then oogenesis on the female side, it's the production of that egg inside the ovaries, right? And this is, as I mentioned, a great big giant single cell that gets fertilized by the sperm, okay? So obviously hormones play a role in the production of both of these gametes, and they are essential steps toward the fertilization process. Okay, so how does fertilization work? Well, you have the fusion of the egg and the sperm to form the diploid zygote, right? The zygote, that sort of pre-embryonic combined cell. And it's kind of the very first step in developing a new organism, right? So the process starts with recognition and binding. So there are receptors on the outside of the egg cell that the sperm has to like attach to, almost like a lock and key fit. Okay, so there is binding to the receptors. That's when the acrosome reaction takes place. So the head of the sperm sort of breaks down some enzymes, helps it sort of penetrate through the cell membrane of the egg. Membrane fusion, okay, so when the membranes of these two cells fuse together, the nucleus of the sperm can enter the nucleus of the egg, and you now have this combined material between the two cells, and then the egg is activated, right? And so the cell begins to prepare to divide and grow, and that is the egg that has been fertilized by the sperm. Okay, so these are those sort of steps kind of mapped out for you going from, let's say, bottom left, and then in a sort of clockwise fashion around to the right. Okay, so it starts with the acrosome reaction after binding, I should say. So there's binding of the sperm to the receptors on the egg cell membrane. The acrosome reaction breaks down some enzymes. That those little balls on the outside are part of a layer called the zona pellucida. That's like a protective coating that protects the egg, and it sort of selectively lets the sperm in. And then you have membrane fusion, the nuclei combine, and you've got this cell, this diploid zygote, ready to rock and develop and split. Okay, so that now the next step is, some pretty intensive, rapid cell division, right? So you get this like splitting and growth of our zygote and our embryo. And so there are different steps to that process as well, right? So you're taking something from a single celled zygote into this embryo. The first step is cleavage. So if you can imagine, the egg has been fertilized by the sperm, it's a zygote now, and you get massive and rapid splitting of cells within the original cell. So it's like rapid cell division, but you don't see any growth in the size of the egg at the beginning. Okay, so that is cleavage. Okay, and at the end of cleavage, the structure is just a whole mass of number of cells called the blastula. And then once you form the blastula, you get the process of gastrulation, where um, the cells begin to reorganize themselves into layers. We call them germ layers. You might want to call them like dermal layers. Basically, different sort of source material, okay, that is going to lead to the development of the different organs in the body. Okay, so gastrulation is this kind of like reorganization and specialization of our cells into layers. Okay, so from the blastula, you have gastrulation, and then ultimately it's organogenesis. And you can picture that, that ultrasound with this little baby in someone's belly that looks kind of like a fish or an axolotl. I don't know. And you now have the formation of like distinctive tissues, right? Different parts of the body start to form, arising from these germ layers that develop during gastrulation. Okay, so now you start to get this little heart and this brain and different things sort of developing within the embryo. Amazing stuff. Okay, so here's the illustration of this process, right? So the early stages, the cleavage, as you can see, you go from like this like single-celled zygote to this blastula, which is loads and loads and loads of cells. But as you can see, it's like the same size, right? It didn't grow; it just kind of split and split and split. Now you've got a whole number of cells which are able to reorganize themselves through gastrulation. Now you've got different layers, differentiated tissues within, and that will lead to the formation of the organs. Okay. And ultimately, we get this development from embryo into an adult, right? So continued differentiation and growth, specialization, right? Cells take on different specific functions. You have like these somatic cells. You have these like undifferentiated cells that are the source of new physical material that will be differentiated in different organs of the body, different structures, different adaptations. Okay, an important point on metamorphosis. So this is something we see in some of the animal world, right? You can have sort of different degrees of metamorphosis, right? You have sort of gradual metamorphosis, or you can have like complete metamorphosis. All right. And so some of the obvious vertebrates we can think of are the amphibians, right? And the salamanders, you often get these aquatic larvae with external gills that crawl out, right, and become these adult salamanders that we recognize. Sometimes throughout the organism's life, the organism retains juvenile features. So the axolotl I mentioned, right? They live their lives looking like larval salamanders. Pretty amazing stuff. You think of frogs, right? Going from egg to tadpole to adult frog. That's pretty big metamorphosis. And then in the insects, we see this complete metamorphosis where actually there's a pupal stage, right? So this intermediate stage between the juvenile and the adult, the larva and the adult, where it huddles down into this cocoon-like structure, right? And undergoes like a full-on transformation. All right. And then on the other end of the scale, after our sort of initial fertilization and growth and development, ultimately, we're all going to die, right? So we call that senescence, right? So the gradual, like sort of slowing down, deterioration of our functional abilities, right? Everything starts to work less and less well. All right. It's all kind of a function of aging and our cells and our genetic material aging over time. We become more vulnerable to disease. We have this telomere shortening that we talked about, right? Our DNA starts to shorten, it gets trimmed on the ends, right? These tag ends that we have in our DNA reproduction. And then with the accumulation of these free radicals, these reactive oxygens in our body, then you get some more damage to your cells, which leads to more mutations and more breakdown. I mean, it's not to be too morbid, but this is just part of getting older. Your overall sort of biological function starts to deteriorate a little bit. We have important protections in place, and we've got genes like P53 that regulate, cell death, right? So through apoptosis, programmed cell death, we've got some that'll activate, that'll kick into gear and say, okay, this line of cells is getting old. We're prone to mutations, we're prone to errors and degradation now. Let's knock this line out, and we'll focus on the development of fresh cell lines within the body. Okay. And when we look at aging and sort of breakdown, there's sort of two schools of thought, right? There's the idea of genetic programming of aging, right? Where, it's like your own genetic material has sort of a shelf life to it, right? So it's kind of encoded in the organism how long that organism is going to stick around. And then there's the damage accumulation theory, which is more sort of the nurture, if you will, right? Nature versus nurture debate. That's like environmental accumulation of damage over time as well. Okay, so I think most of the scientific community would agree that this is something that where both of these theories play a role as we sort of age as organisms. Okay, it's important to recognize that apoptosis plays a critical role in sort of fighting against cancer and other diseases where cell mutations and cell damage over time is so arrested or cut off by this pre-programming of gene activation that shuts down cell lines before they become problematic. Okay, so just quickly, we'll wrap up with our case study, and this would have led to our in-class exercise. This week's in-class exercise was pretty basic. We were just going to observe the different life stages of our friends, the mealworms, the pupae, the adults, and the larvae, and we're just going to maybe talk a little bit about sort of, the advantages and disadvantages of complete metamorphosis. Here's what I would say. So with insects, we have sort of this gradual growth. It's a metamorphosis that's more sort of incomplete. Okay, and if you think of something like a grasshopper, if you see a grasshopper nymph, okay, a juvenile grasshopper, it kind of looks just like the adult without wings. Okay, and then over time, it'll molt 5-6 times throughout its life and develop those nice long wings you see on an adult grasshopper. It'll get bigger, right? And of course, because they have an exoskeleton, they need to molt in order to grow. So they pop out of their, external shell and grow into a larger, fresh one. So most insects grow in that way, and we call those hemimetabolous insects. They just become like sort of bigger, more winged versions of themselves as they get older. But some of the key groups, and I would refer to these as the big four orders of insects. So order, taxonomic level order. There are four which have the greatest diversity on Earth, and those are the Lepidoptera, butterflies and moths, the Coleoptera, the beetles, the Diptera, flies, and then the Hymenoptera, which are wasps and bees and ants and things like that. Those four orders of insects in particular undergo what we call holometabolous development or holometabolous metamorphosis, where they go through this complete transformation and there's a pupal stage involved. So the juveniles are not just smaller versions of the adults without wings, okay? They can be completely different. They can have completely different life strategies, and they can occupy completely different ecological niches. So I'll take like a hoverfly, for example. This is a group of flies, so in the order Diptera, in the family Syrphidae, we've got these beautiful little flies. They hover around flowers like helicopters, and a lot of them are bee mimics. They've got, or wasp mimics. They've got the black and white or black and yellow striping. And no doubt you would have seen these if you've been in any sort of areas where there are gardens and flowers. Okay, so pollen feeding adults. They're mimicking bees so they don't get eaten by too many birds. And they're really kind of just sort of look like a fuzzy bumblebee sometimes. Um, the larvae, on the other hand, are voracious predators of aphids. So on those very same plants where you see the adults feeding on nectar, the larvae are aggressive predators. So occupying this different sort of level in the food chain, right? There's different space in the ecosystem. It's an interesting strategy, because these different life stages are obviously functioning in very different ways. Okay, so there is this important hormone, the juvenile hormone, that maintains larval characteristics until such time that ecdysone is expressed. Okay, so another hormone that says, okay, time to molt, and then time to develop into a pupa, and then from the pupa into the adult. So just a couple of the hormones that play a role in metabolic development and metamorphosis in the insects. Okay, folks, so we are not going to complete the weekly lab activity. Obviously, we're not together in lab for week 13. And so those 2%, that 2% mark for the lab activity will just be distributed across the students' marks for the second half of the term. So hopefully that suits everybody. Thanks for tuning in. I know it's not quite as exciting when we're doing this virtually. So sending good vibes, wishing everyone all the best, and looking forward to connecting with you in class in week 14. All right. So take care, everybody. Bye for now, and we'll see you soon. Cheers.